Hello my dear students. Today in this very part of chapter number 2 we'll study about the microsporogenesis, means how the formation of microspores, means the male gametes required for the sexual reproduction in the flowering plants, takes place. And along with that we'll study the structure of those gametes, which will be the pollen grains, and how those pollen grain forms the pollen tube, and how those pollen tube reaches towards the embryo sac. So in this very part we'll study the process of formation of pollen grains, as well as the structure of the pollen grains and how the pollen tube formation takes place. So first of all, before moving towards the process, we must know about the word means microsporogenesis. What does this word actually defines? See, this microsporogenesis is made up of two words, that is, if we talk microspore and genesis. Although it is written microsporogenesis, so consider this word as microspore and this is the genesis. So it is made up of two words separately, and if you go through the meaning separately, you will find that microspores, here is the microspores, and the genesis means formation. So the formation of pollen grain is refers to as microsporogenesis. And what are these microspores and how these microspores convert into pollen grains, we'll study in this process. So first of all, the process will go through the points. First of all, we'll study the theoretical, and then I'll show you the figures, how those flow chart, how those formation of microspore takes place. So see, the formation of microspores inside the macrosporangia, or the pollen sacs of the seed plants. The formation of microspores takes place inside the macrosporangia, and these microsporangias are also refers to as pollen sacs. When we have studied the longitudinal section of anther, we have gone through the pollen sacs and microsporangia. So microsporangia also refers to as the pollen sacs, which holds the pollen grains that are formed in the seed plants. And the second point, if you go through, we'll see that it is a diploid cell in the microsporangium. Actually, if we talk about the ploidy, means first from the diploid cell the gametes formed will be the haploid. So before undergoing the process, the cells will be diploid in nature. So the diploid cell in the microsporangium, which is called as the microsporocytes or a pollen mother cells, it will undergo meiosis, and then it will give rise to the 4 haploid microspores. So first of all, those cells will be diploid in nature inside, when they'll be inside the microsporangia, and those diploid cells inside the microsporangiums are either refers to as microsporocyte, it is written here microsporocyte, or PMC, it is refers to as PMC, pollen mother cell. Sometimes it is written in this abbreviated form, so you must know what PMC means. So PMC means pollen mother cells. It is also known as microsporocyte. So never confused with the words like microsporocytes and pollen mother cells. Both are the same things. These cells present inside the microsporangium undergo some differential changes that leads to the process of meiosis, and that meiosis will give rise to the 4 haploid microspores, and those 4 haploid microspores will be the pollen grains, will convert into the pollen grains, and those pollen grains will be the male gametes required in the sexual reproduction in the flowering plants. Now these are some of the detailed points about microsporogenesis. So the first point is the process of formation of microspores from a pollen mother cells through meiosis is called microsporogenesis. This is one of the most defined way of describing the microsporogenesis. So if you have to describe it in a very short way, so that you can go through this very point, and this will be the most appropriate way to explain the microsporogenesis. The process of formation of microspores from a pollen mother cell through meiosis is called microsporogenesis. The second point, the cells of the sporogenous tissues undergo meiosis to form microspores tetrad. I have told you there are cells inside the microsporangium, means the cells which are present towards the center of the cell of microsporangium are called as the sporogenous tissues, and those sporogenous tissues have some kinds of the cells. These are made up of a group of cells, that's why they are referred to as sporogenous tissues. And those groups of cells which is refers to as sporogenous tissues undergoes meiotic divisions to form microspore tetrads, and these microspore tetrads on dehydration are arranged. Microspore tetrads means they are arranged in a tetradal arrangement and clusters of 4 cells, as the word itself tetrads tells you. And these microspore tetrads, upon some kind of dehydration condition or maturation conditions, separate themselves, and each of 4 microspores, now they have become an you can call free microspores. Those free microspores are now free to convert into the pollen grain. And as each of the sporogenous tissues has a potential, I have told you each and every cell of those microspore tetrads which are arranged in a tetrahedral forms have the capacity to work as PMC. They all, the cells which are present inside the sporogenous tissues, each and every cells present inside the sporogenous tissues have the capacity to behave as PMC, means pollen mother cells, and these pollen mother cells will undergo meiosis to form the microspores. And first of all they will be in tetrad form, and then on the dehydration they will be released as dehydration, they will be released into the atmosphere and develop into the pollen, mature pollen grains, and pollen grains with the dehiscence of anther, and those dehiscence line of dehiscence, through that line of dehiscence that pollen grains will be released, as we have seen the line of dehiscence in the longitudinal section of anther. Now this is a very well defined way to show your process of microsporogenesis by this flow chart. So this is a most appropriate way if you don't have to make a diagram or you are not good in diagrams. So this will be the best way to explain the process, and the most scoring way to in your board exams also, most scoring way to explain the process of microsporogenesis. First of all, I have told you that the microsporogenesis, thus if we have gone through this, if you recall the structure of the anther walls, means we have gone through the LS of the anther. So we have seen it is made up of 4 layers, means anther wall is made up of the 4 layers, and those 4 layers are epidermis and endodermis, middle layer and tapetal layers. And out of those layers there is another and those layers surrounds a sporogenous tissues present in the microsporangium, and that sporogenous tissues have a number of cells, and though each and every cells present inside the center of the microsporangium is known as sporogenous tissues, and those sporogenous tissues are made up of a group of cells in which each and every cells have a capacity to convert into pollen mother cells. And the sporogenous tissue, if the ploidy has been asked about sporogenous tissue, so you must know about the ploidy of the sporogenous tissues. It is always diploid. So the conversion of microsporogenesis is from diploid to haploid, means the process will go from the diploid number of cells to the haploid number of cells, means from PMC that will be diploid in nature, and the pollen that will be formed will be haploid in nature. So each and every cell that will be present inside the sporogenous tissues will act as the pollen mother cells, and each and every cells of the each and every cells will undergo the process known as meiosis, and this meiosis you already know, it is refers to as reductional division. So in this the number of cells will be reduced, so from 2n they will be converted into n, half the number of cells will half, and this process of converting the pollen mother cells into the microspore tetrad is known as microsporogenesis. And now after reductional division, undergoing one first meiotic division, second meiotic division, it will form a microspore tetrads. So after forming the microspore tetradal arrangement, it may be in any kind of arrangement, it may be isobilateral arrangement, it may be tetrad, it may be linear in arrangement. So depends upon the this microspore arrangement depends upon species to species of the flowering plant. So need not to worry about that, the microspores are always arranged in a tetrahedral form. So they can be arranged in the isobilateral form, they can be in linear form. So need not to worry about in which form the arrangement is. And after the microspore tetrad formation has been taken place, each and every microspore tetrads will be diploid in nature, already written here. Now those microspore tetrads, upon maturation and dehydration, these will dissociate from each other and matures into the pollen grain. See here, this is a microspore tetrads. First of all, when they will be formed, they will be in such a cluster arrangement, but when they once get matured, they will be released out. They will be released out as a free pollen grain, mature pollen grain with several structural changes in them, and those structural study will do in the later slides. See the microsporogenesis, how the microsporogenesis development of the pollen grains takes place. First of all, I have told you that there is a mass of an undifferentiated cells, means those mass of undifferentiated cells, if we recall the structure of anther, um, if we recall the structure of anther, so we know that it is made up of the 4 layers of the cells, and those out of those 4 layers of the cells, the outer layer of the, how those outer layer of the cell wall formed, and how those the cells inside the sporogenous tissues, cells towards the center are formed. So this is the way to explain the process of the pollen grain formation. First of all, these mass of undifferentiated cells, which is known as in immature pollen grain, if we go through, we'll find that and mature pollen in the all mature anther, if you see the these you will find that these are undifferentiated mass of cells, means you cannot differentiate each and every cell from other. So that is a homogeneous mass of a cells, and that homogeneous mass of a cells upon maturation, when they attain some kind of maturity, what happens, the cells of some of the cells of this homogeneous mass becomes cytoplasmically dense, becomes cytoplasmically dense. After becoming cytoplasmically dense, they are referred to as archisporial cell. Archisporial cell formation you can call. So they are referred to as archisporial cells. Now these archisporial cells will divide periclinally, and after dividing I will tell you about periclinal and anticlinal. So need not to worry about what does this terms periclinal and anticlinal came from. So I'll tell you all about periclinal and anticlinal. So archisporial cells, after formation of the archisporial cells, now these archisporial cells will divide periclinally. So after dividing periclinally, first will be the first periclinal, after dividing periclinally, that it will divide into 2 layers of cells. The very first layer will be the outermost layer, which is refers to as the primary parietal cells, and the second layer will be the primary sporogenous cells, which will be below the primary parietal cells. So these primary parietal cells will again undergo the anticlinal and periclinal division. Before so first of all, it will grow anticlinal divisions, in which the number of cells will increase. Increasing the number of cells will lead to the formation of endothecium, middle layer, and tapetal layer. Means here you have found the number of increase in the number of cells as well as increase in the number of layers. So primary parietal cells will lead to the formation of the layers of the cells, means the layer wall, anther wall layers, and the other form we have got from the archisporial cell periclinal division is the primary sporogenous cells. These cells are present towards the periphery, and from the periphery it leads towards the layers, and after leading towards the layer, when we come to the center of the anther structure of the anther, longitudinal section of the anther, you will find up sporogenous cells, and those sporogenous cells are present towards the center. Means these sporogenous cells are present towards the center, and those sporogenous cells will have a kind of a cells, each and every cells of those sporogenous cells will undergo mitotic divisions, and those mitotic division will lead to the formation of the MMC, and after undergoing mitotic division, they will lead to the meiosis, and meiosis will lead to the formation of pollen grain. So each and every cells present inside the sporogenous cells will act as a microspore mother cell or pollen mother cells, and those pollen mother cells or microspore mother cells will undergo meiosis or reductional division. So they will form a microspore tetrads, and those microspore tetrads on maturity will form the pollen grain. So this was all about how the pollen grain formation takes place. Now Let's see about what is periclanal and what is anticlinal division. If see, this is, if you are going through this figure, I have taken an initial stage of leaf formation in which the epidermis is being divided into periclinally and anticlinally. This periclinal division shows you the increase in the number of layers. This red line is showing you the increase in the number of layers of the cells, and this blue line which shows you the anticlinal divisions. This increases the number of layers and this anticlinal division, if you see the blue line here, this is increasing the mass of a cell. This is increasing mass of cell. Got it? So periclinal division, just you have to know about only one thing, that periclinal division increases the number of layers and anticlinal division increases the mass of the cells. So here you can find the mass of the cell is increasing, and here we'll find there is a number of increase in the number of layers. So this is all about periclinal and anticlinal division, and nothing more to know about periclinal and anticlinal divisions. Just clear with the concepts of periclinal and anticlinal division is this very much important to know. If we talk about how the pollen tube formation or how those tetrads tetrads releases into the free microspores, and then those microspores undergoes mitosis 1, 2 and leads to the formation of vegetative and generative nucleus. So this is a flow diagram to show you the how the pollen grains are released from microspore mother cells to the pollen grain formation, and from pollen grain to pollen tube formation. See, first of all, this is a structure of anther, and these are all the outer layers. These are all the outer layers, and this is a sporogenous tissues. And these sporogenous tissues have a number of cells. This is number of cells, and each and every cells inside the sporogenous tissues are capable to convert into microsporocyte. This is a microsporocyte. And this wall layer which provide nourishment to the developing embryo, means the layer which is present inside the sporogenous tissues, means very much close to the sporogenous tissues, is tapetal layer. Got it? So this, all those cells present inside the sporogenous tissues or in a group of tissues, those cells are capable of converting, are capable to form a microsporocyte, which is also called as or PMC. Now these PMCs will undergo meiosis. First of all, they are diploid in number 2n. They'll undergo meiosis, means they'll undergo reductional division. So, if first when they go under meiosis 1, they will lead to the formation of the two microspores, okay? Two microspore formation, and those will be haploid in number, and then they'll undergo meiotic division 2, which will lead to the formation of 4 microspores, and those 4 microspores will arranged in a tetrahedral arrangement. This is a tetrad arrangement, and upon maturation they will be released as a microspore, and these microspores are the mature pollen grains, and those mature pollen grains, after getting mature, they will convert into pollen grains, and those pollen will undergoes mitosis 1, and they contain this is a mature pollen grain, and these mature pollen grains have an unequal division of cytoplasmic division, nuclear division, and those unequal division will lead to the formation of a one large cell and one small cell. So this will be the one large cell vegetative and the small generative cell will be formed. After undergoing mitosis 1, they will form one vegetative and another generative cell. And after forming one generative and one vegetative cell, this will now it is now a two-cell structure. Pollen, which is a two-cell structure, now it will undergo pollen mitosis 2, in which this vegetative nucleus will no more divide. It will no more divide, but the generative cell nucleus will divide again, and means it will undergo mitosis 2 to form the two male gamete nuclei. Two male gamete nuclei will be formed, and those two male gamete nuclei will be formed, and after forming the two male gamete nuclei, this will be called as a mature pollen grain. This is a mature pollen grain, and after this you are seeing this wall around it. This wall around it has an outer wall of the pollen called as outer wall, called as exine, and this is having a space. These spaces are called the germ pores, where the exine formation do not takes place, and These will lead to the formation of the germ pore, and those germ pore will lead to the formation of the pollen tube. And those pollen tube will carry out the two male gamete nuclei, and along with a with vegetative nucleus, which is also called as the tube nucleus, they will lead towards the embryo sac. See, they will when they will lead towards the embryo sac, always the tube nucleus or vegetative nucleus will be towards the front, and the sperm cells, the male gamete nuclei, will be towards the back side. What does this mean? Why they are towards the back side, and vegetative nucleus, which is of no importance, is towards the forward, is forward. So the this vegetative nucleus is towards forward because it guides these two male gamete nuclei to reach towards the embryo sac. So this is the importance of tube nucleus, to be forward before the male gamete cells or sperm cells. This is all about how the microsporocytes convert into the microspores and microspore tetrads and leads to the pollen grain formation, and those for pollen grains into vegetative and generative cells. This is all about the pollen tube. This is your pollen mother cell. This microsporocyte is the pollen mother cell. This is PMC, which will be diploid in number, will undergo meiosis 1 to form the 2 haploid microspores, and after undergoing meiosis 2 it will form 4 haploid microspores, and this will arranged into tetrad form, and this tetrad forms upon dehydration will release into a microspores, and released microspores have a polarized nucleus, and these polarized nucleus will undergo pollen mitosis 1, where it will be divided into vegetative nucleus and a generative nucleus, and after this the vegetative nucleus will no more divide, means till then it remains as a two-cell stage, means it will contain a generative cell and a vegetative cell. Up to this stage there will be two cells, means vegetative and generative cells. After undergoing pollen mitosis 2, they will converted into a tricellular pollen or three-cell pollen stage, which is in which the vegetative nucleus will remain as such with no further mitotic division, but this generative cell will undergo mitosis 2 to form the two sperm cells or male gamete nuclei. This is refers to as a tricellular pollen. So this is a mature pollen grain and it's a tricellular stage. So this is all about the development of pollen grain we have talked about. Now we see the structure of that pollen grain. The pollen grain, first of all, is refers to as immature male gametophyte. Immature male gametophyte is the first point of the pollen grain to be remembered always. And this outer layer of the pollen grain is always thick, rigid, and ornamented, which is called as the exine. This is the exine. You are seeing this is very much thick and ornamented, like spike-like structures you will find here. And these exine is formed by cutin and sporopollenin. This a very important question which is always being asked in competitive exams also, as well as can be asked in your board exams also. What is the exine made of? What this means the exine is made up of? So you will have to answer, means exine is made up of sporopollenin. If you don't tell about cutin, it is nothing happens, but if you don't tell about sporopollenin, it is very much important to tell about that the outer layer of the pollen wall is known as exine, is made up of sporopollenin. So always remember this sporopollenin. Sporopollenin is a high resistant material and is non-biodegradable. Sporopollenin is a highly resistant and material, means it is resistant during the adverse conditions, and it is non-biodegradable. That is why, being non-biodegradable, the fossils of pollen grains have been reported, and those pollen grains of fossil pollen grains are referred to as palinofossils. And this internal layer, if we talk about the internal layer, the second layer, it is called as intine. This intine is very soft, thin and elastic, which will lead to the formation of germ pore. And this thin layer is made up of pectin and cellulose. These germ pores are initiated from this inner layer, which is intine, and these germ pore will lead to the formation. These are the sites where the exines have been degraded, and these degraded sites leads to the formation of the germ pore, and these germ pore will form the pollen tube. So this is the structure of pollen grain. You can answer very shortly and very correctly in your board exams also. How there is a pollen kit formation also takes place in some of the plant species. So this pollen, what is the significance of this pollen kit? We'll have to study only this much about this. First of all, the plants in which pollination takes place by insects, their pollen grains having oily layer around them, it is called pollen kit. In those plants in which the medium of pollination is by insects, their pollen grains having only oily layer around them, it is called pollen kit. And you can see here the structure of a pollen kit. This is a typical structure of a pollen kit shown here. Typical structure of a pollen grain, and this is the fatty substances, that oily substances that forms inside this, and these oily substances are fatty substances, and these fatty substances are made up of lipids and carotenoids, and this is a sporopollenin. This layer is the sporopollenin. This content of the outer layer is sporopollenin, which the importance is that it provides protection from harmful UV rays. This also helps the insects to get sticked with the pollen grains, and when they stick to the pollen grain, they can reach to the stigma of the female flower so that the pollination may takes place. And they being yellow in color is to attract the insects to come and pollen grains to get attached with them. So one of the best examples of pollen kit formation has been told in NCERT is of Cypsela. So if Cypsela is the referred plant in the NCERT. So if the example of pollen kit has been formed, or pollen kit has been found in which plant, give an example. So you will have to answer Cypsela, as this is a referred plant in your NCERT. I have already told you all about this, so need not to waste my time on this very slide. So there are some important points about pollen grain you must know. So first of all, these pollen grains being well preserved just because of the presence of the very high resistant material on the outer layer of the wall, that is sporopollenin, and because of being highly resistant, they have been kept as fossils. So they are refers to as pallenofossils, and these pallenofossils are they actually helps in the exploration of the sites of petroleum. Pollen grains of many species causes severe allergic. They are very much allergic, and this causes the respiratory disorders like asthma and bronchitis. So there is a Parthenium which is known as the carrot grass, which is called as invasive species, came from foreign, came to India as a contaminant with imported wheat. One of the pollen grains of Parthium is very much allergic. So it has become an ubiquitous in occurrence and causes pollen allergy. Pollen grains are rich in nutrients, taken as a food supplements as well. Pollen banks has been created in order to keep those pollen grains for the fertilization to take place. For means if you have to do a fertilization for of a respective species of a plant, so you must have to keep that pollen grains, so that those pollen grains we can use in later on. So keeping that pollen grains, we have made a pollen banks for the future perspective. So those pollen banks keeps those pollen grains in a liquid nitrogen, which is near about at a temperature of -196 degrees Celsius, and this process of conserving pollen grain is refers to as cryopreservation. This cryopreservation has been asked in short notes, so remember these points about the pollen grains. So this was all about the microsporogenesis, structure of pollen grain, and pollen tube formation. We will talk about the next topic very soon about megasporogenesis. Till then, thank you.